So I thought, so we did receive some feedback that there was a bit of a lack of overview on spatial biology. We were too focused on the tech the last time. So I've kind of tried to put together like a panorama of what is happening out there with a special focus on spatial transcriptomics, because if we talked about all the other modalities, we can talk till tomorrow. Okay. No. Okay. All right. So this is going to be something of an interactive talk. So feel free to stop me if you want to ask any questions. I'll try my best to have time in between. So this is like a list of content for this talk. So some of the things that I find that I think is important is to talk about exactly what is spatial. All right. Knowing the spatial diaspora. All right. So what are the current technologies, the platforms, what's fad, what's fiction, and what's reality, right? So there's a lot of platforms out there, and it's difficult to choose and it can be confusing. And then you want to understand what's your platform potential, as Trevor mentioned, its limitations, and also its open access capacity. And I'm not talking about computational method analysis pipelines that you find on GitHub. And then we'll broach on computational diversity, development, and growth, and also talk about wet lab and engineering expertise, because these actually are the foundations for all your platforms and your computational pipelines. And then, of course, what this workshop will teach you and your approach to this course, which is, of course, very individual, but it's just to let you know that you are the drivers. We are just facilitating every single one of you here. So what is spatial? So common thing is spatial single cell. So now everyone went through that whole craze of single cell, and it was very valuable. And then is the next step spatial? So is spatial just an advancement of single cell? Is it only protein and gene expression? And why do you want to do spatial? Is it because you want to understand cell-cell communication? And is it better than bulk or single cell feature extraction? Is it something that is just trying to improve upon previous development, or is it a complementary tool? So no, it is not single cell only. It is actually the study of collective relationships, right? So that is why we do spatial. We want to have that, adjacency matrix. We want to have a snapshot understanding of what your neighborhood is. So it can be long distance, short distance. It does not necessarily have to be single cell. It may or may not be molecular in nature. So why this statement? You don't have to understand your gene expression, your transcripts, your protein, or your metabolites. You can just look at the centroids of your cells and look at their geometries. That is also spatial, and that is what a lot of plant scientists and insect biologists do. And that's what geospatial analysis is all about. That's how cities come about. And is it just cell-cell communication? I think CC is just one task within the broader area of tasks that we can apply over spatial. And it simply gives a different vantage point, at least in my opinion. So it's not about being better than bulk or single cell. I think it really depends on the depth of the question. So sometimes if you have to so all these technologies have their limitations. So you want to have something that's more granular and fine-tuned, then go for spatial and single cell. If you want to have something that is more in-depth, quick, and also that is more, reliable, then I think in many cases, bulk sequencing still helps as well. Now, knowing the spatial diaspora. So this is the part where I'm just going to talk about what spatial and how the technology developments have been about. So we're going to focus on transcriptomics. So basically, the whole premise of all of these platforms is to capture the molecules that govern the central dogma. So everyone we would have learned the central dogma. You have DNA, and then it goes to form your RNA, and then you have your proteins, right? And then all of these features, molecular features, you have your spatial dynamics, you have your temporal dynamics, and they govern biology and many aspects of biology, not just mammalian, also insect and plant, and also in healthcare. And then there is the multidimensional understanding of cells. So the basic essence, the engineering principles that drove spatial technology development is to understand everything in context of neighborhoods. So broadly speaking, you have your sequencing based and you have your imaging based techniques. Now, the foundations, the chemistry, and the engineering rules that actually help to develop these platforms will affect your computational analysis, your choice of methods, and also help in answering. It will actually guide you if you can answer your biological question. So just a brief overview. So we will try our best, or at least towards the end, to also give you an understanding of the differences in sequencing based and single molecular based. So for just a brief overview for those who are not who are new, all right. So imaging based is what it literally means. It's using a microscope. So you have microscope, so you have a tissue, you label it, you label your transcripts with probes. These probes are tagged with fluorescent compounds, and then you have a microscope. These microscopes have a resolution of 200, 250 nanometers, some of them 300 nanometers, and then there is subcellular resolution through computational methods, and you get spot-based transcriptomic data set. So basically, you have a data frame. Everything is a data frame, and everything is a matrix, basically. So you have your X, Y coordinates, and each is a transcript point. It is something that people have done in fluorescence microscopy with protein antibody labeling. It's not something that is new. And then you have sequencing based. Sequencing based, as what it implies, is you are sequencing out all this information. And they can be probe-based, they can be whole transcriptome, but these rely on already bar-coded arrays of slides. Basically, these are spots. So the thing is, you are not interrogating single cells. You are interrogating spots. Each spot has got a barcode array, and the mRNA is sequenced, and you're capturing that. And so how and whether and if you segment or not depends on the size of these spots and the nature of whether it is a transcript or it is basically an integrated spot, right? So all of this has to go. So the thing is, understand your tech. So just some principles, right? So for instance, just to drive in this point, so Curious Seeker is the company that was started and born out of the platform, the technology called slide decks. Many of you would have read that. So it's a really cool technique. It's using beads, basically. So you have beads that are tagged to barcodes, all right? And these barcodes will be released and they will attach themselves to the mRNA from the tissue that is overlaid on these slides. Now, this is very much nuclear specific. You tag your nuclei, and then you isolate these nuclei, you get a bunch of beads, basically, and then you run them through a microfluidics chip. All right, so it kind of in many ways, it resembles your traditional single cell fluidics technology. Now, Visium HD is different, right? So Visium HD together with StereoSeq. These are array-based. So you have your oligonucleotides, which are imprinted onto these spots. The size of these spots do matter. And the probe chemistry also matters. So for instance, in Visium HD, you have barcode arrays which are imprinted. StereoSeq, it is what is imprinted. Imprinted is these are actually nanoball oligonucleotides that are imprinted. So you have your RCA nanoballs that are already on the slides, and also the dimensions of your spot is different. There is no space in Visium HD. There is dead space in your stereosig. So the resolution for stereosig is better. It's 500 nanometers as compared to 2 micron, but there is dead space. So that's all this. So the actual size of the spot is about 250 nanometers, I think. But it changes with the chip. All right. So the thing is, why does all this matter? It's not just about drawing segmentation. You will also face another issue when you start to go deep into your analysis, and that is RNA diffusion and how you process your tissue. What kind of tissues will benefit from this tag, especially from tissues, for example, that have very loose and free RNA. And then you have your imaging-based platforms. So I'm just going to put through, I'm giving all the typical classical examples here. These are not the only platforms. These are probably the ones that you would have heard about, and it's the most popular. So we have Xenium, and we have Cosmax, and Immerscope. These were the three primary industry players in single molecule microscopy imaging of transcripts. So for the Xenium, you have your padlock probes, and the padlock probes and the RCA amplification really helps to improve the sensitivity of the system. CosMX actually came out with their single molecular hybridization technology first before, and they kind of use sort of what we call linear encoding and decoding system. So it's you have a linear array of probes that attach themselves to your transcripts, and these are amplified. Now, Merscope Merscope is important, at least to me. So one of it is the whole encoding and decoding process is unique, and it is also robust to error. But what is actually more important when it comes to Merscope is they're probably the only ones where their technology baseline and all the probes and the way they decode and encode their image, the whole process is actually open access. So why is this important? You're not seeing these probes. These probes are interrogating your transcripts, right? And these are basically, just imagine that your microscopes are 250 nanometer resolution microscopes. But these platforms are giving you transcripts that are subcellular, that are 100 to 30 to 100 nanometer resolved transcript spot sized. So it's a probabilistic model. So it's to do with your point sources of light, and they deconvolute that. So when a platform is open source and they deliver and they give you all the raw images that comes out of that microscope, you actually have the opportunity to enhance the algorithm in predicting your spots. And even though this is something that we don't talk about, but there is a scientific community that actually does this sort of things all the time. So sometimes it helps. So it is a balance between what is convenient, all right, and how much you really need to know. Sometimes we don't need to know that much of details, and sometimes we do need to understand the merits of the platform. So everyone has their own right, to preserve and to hold back. But understanding all this will kind of make you understand what are the different platforms, and also how unique and what you can do with them, and how much you can grow from it. So these are all pre-commercial, and PixelSig, it sounded very complicated when I read this, but it was really nice. So what PixelSig does is you have an array which is you have a slide, and you basically have an array of barcodes, and you can use it to stamp. So you can actually replicate your barcodes. And how it replicates is you kind of have this set of specialized enzymes where when you overlay another slide over it, it kind of copies over the barcode. And I am not sure personally how this is going to be effective. It sounds very tedious, but it's an interesting read because it's basically about stamping. It's like you're taking a DNA stamp and then you're stamping it on many different slides. And then singular genomics is very similar to Xenium, very, very similar to Xenium, but they already seem to have a protein panel that is integrated with the transcript interrogation as well. And then what I really wanted to show, and someone talked about 3D, is stellaromics. So they are pre-commercial, they just got their funding, but that is the potential of their platform. So the field is truly advancing very fast. And stellaromics is interesting because I read this paper a while ago and then I completely forgot about it. And then I read about them and then I realized that this was a science paper in 2018. And I'm going to talk about this paper later on. So I have not covered every single spatial transcriptomics platform. And this lecture basically excludes other spatial modalities. So I'm not talking about the metabolites or the protein or the epigenomics or even about temporal spatial patterning. All right. But this is just one avenue. And you have right now, you have platforms that are coming with in situ proximity ligation at the single cell level, at subcellular levels. So the field is really advancing. And the more you know about what is out there, you don't have to know everything, but if you know in general what is out there, I think it will help guide you in your research questions. So yeah, so sometimes the platforms are similar, same design, same underlying principles. Seems like, it's low diversity. So how do you choose then? I think sometimes we have to make practical choices. It is true that some vendors tend to be more approachable when it comes to how they package, all right? So having all this information will help you. And also sometimes the underlying principles are similar, but chemistry and engineering differences can contribute to differences. So there was once it was just a random observation that I had, is that when I did a MERSCOPE analysis and a Xenium analysis of the same tissue, I couldn't detect differences in drug treatment in my Xenium analyzed data set, but I could actually capture the difference in my MERFISH data set. And the difference is maybe likely due to the fact is that you have this amplified transcript pool in your Xenium. You don't get that in MERFISH because it's linear encoding. It's not amplifying, it's not geometric, right? So sometimes the platform and the chemistry and the way something is designed to capture a signal, all right, can make all the difference in answering your biology. And it's the same for segmentation. When Bayes-Saw first came out, I like Bayes-Saw in general. So Bayes-Saw is a segmentation. It's a statistically powered segmentation. It's unsupervised. It works very, very well in most platforms, but there's a bit of fine tuning on certain platforms. And I think this has got to do with to take into account the number of transcripts, the number of genes, the density of your genes, right? And also think about all these things like there's optical limit, especially for your imaging-based microscope, right? So which means that you cannot push the limits if you want to do copy number variations. You want to do copy number variations, then choose a spot-based spatial transcriptomics platform and not a single molecular imaging-based, unless we can improve on that optical limit and the bandwidth of how much of genes, right? Yeah. So if you understand how your technology works, all right, then it doesn't become like a fad. And what you want is you actually have it, you maximize the potential, and also think hard about older techniques. Does it like just mirroring what Trevor said as well, which I agree. if you don't have to do spatial, all right? So no, let me rephrase that. If you don't need to use a spatial tag to answer your spatial questions, then don't do it. This is a rapidly evolving field, all right? It's a bit scary because every day you read companies eating up other companies. Yeah, and Illumina just announced yesterday that they are going to be having their own spatial platform next year, 2026, which is compatible with NovaSig, blah, blah, blah. So, the field is exploding, right? So understand, always use your biology and your value as a scientist to guide, your truth, your grounding. And then, wait, I just talked about this. Okay, no, I did not. All right. So understanding platform potential. So I'm just going to talk, I think I've mentioned quite a bit already. So what is platform potential? So there are many ways to think about platform potential. It's not just about cost. It's not just about what do you call it, what do you call it, approachable or how good a PR team is. So it's also about how creative some of these platforms are. And sometimes, all right, if you can get the same results, and you see this a lot in imaging-based microscopy systems, right? You have COMET, you have IMC, you have, what do you call it? You have Leica. So the thing is, if you have a Leica microscope and you don't need a COMET, then stick with the Leica because the Leica microscope is incredible. It's probably going to be better than a microscope than your COMET has. If you want high dimensional, then a COMET is worth it. So it's the same thing, all right? So look at the concepts that drive these platforms. Look at the engineering structure. We also have to be practical. If you're not going to get, all right, decent support, then I think it's no point. I mean, research is hard enough. We don't have to vex ourselves with all the tenuous details of having to chase. So these are things about. There are lots of companies out there, but always go with something, with a platform that, I guess I don't want to use the word reputation, but when you talk to them and when you look at their service, do they have the wet lab structure, everything done, and you will know. And then look at the chemistry. I think it's good to look at how the probes work, how the probes are designed. I know that many of these probes are proprietary, they're not going to release, but you can have an understanding, right? You can always go back to old literature and read up on how rolling circle amplification works. You can always go back to previous literature, older literature, and you can work out how, subcellular high resolution microscopy works. Because even though they will boast, and next week is AGPT, they will boast that, how all their platforms are novel, all of this is built on science that has already been done. So you can always get all of this. So do your background check and readings. And some have high sensitivity, but that doesn't mean that they will have good specificity, right? Some of them will have a good PR team. It's always nice to have a good PR team. It makes your life much easier too. And sometimes when you just want to get something going, all right, sometimes you just have to go with something that's convenient, all right? But just know the limitations. Some have convenient and user-friendly packages. And I think that is important, all right? It's not just about packages, it's also about facilities, like core facilities like the Princess Margaret Genomics Center. They provide services, you can rely on them. So reliability is important, and that wet lab baseline is extremely important. Do not take it for granted. All right, and that's it. Some have a more reproducible wet lab processing. There are tons of CROs out there, including the core facilities. All they do is to make sure that the wet lab processing steps are optimized for your tissue. And some are also computationally more tractable. Yes, all these platforms are nice, 2 micron, 250 nanometers, looks nice. But sometimes if it is not, it is just challenging to computationally, manipulate them. So I'm sure some of you would have faced this, how challenging it is to analyze these data sets, right? Just getting them onto a cluster just getting them to run without killing your machines, right, is frustrating enough. So we need to get through these challenges. And for the more advanced users, right, think about, are you developing a method just because you want to show that it works? Or is it really going to be relevant? So if you want to do segmentation, if you want to do, let's say, nearest neighborhood, spatial clustering, the methods, it's not just for the sake of it. It has to be driven also by the principles that form or are they form the foundations of these platforms. So I think at the end of the day, I don't know, I always go back to this thing. You have to understand the science. I think if you understand the science, it will give you a good guide forward. Computational diversity. Okay, so since, this is ISO, it is introductory, all right? So there are a few pipelines out there. So I was a wet lab biologist, and then I started to do computational work, and I'm still learning. And when I first started out, I started with Surat, and it's true, Surat is very easy, so nice, so pretty. Everything looks good. It makes you feel good when you can run your code. So yeah, all this is fine, feels good. But the thing is, I think Surat has got limitations, right? So I feel that for beginners, I would advise you try Surat because it's very intuitive. They have all these very simple functions for the fundamental analysis to be done. They have all these nice plots. You don't have to struggle with any of your plotting functions. So I think it's very good, especially if you want a quick start. We are at an age of the interdisciplinary research, right? We are struggling to learn a new technique, a new skill, and we are also in a time crunch to keep up with research. So that's very practical. I like ScanPy because I like Python. So anything that's Python, I like it. I'm very biased, I know. And I do a lot of image analysis too. But I'm always frustrated with ScanPy and NData and any kind of objects because I feel that I have to put in more effort into it. But I think these two are the basic, but their basic pipeline is not bad, ScanPy. I just feel that it is not as good as you read. That's what I feel. So for beginners, after this workshop or during this workshop, you want to start with Surat first. And then you can try ScanPy because it gives you that Python experience. And I think it is important to know both languages because I think gone are the days where we can just stick with R, we can just stick with Python. For this, in preparation for this workshop, we try to stick to R, and then we are like, my favorite package is in Python. So the thing is, you will find that along the way, you will integrate a lot. And then there's Julia now. And then I'm sure someone will come up with another language with another pretty name. So on and so forth. You don't have to know everything, but I think learning R and being familiar with both R and Python and the basic pipelines from these two languages is important. Now, Giotto Suite. Now, Giotto Suite, I feel it's a little advanced, but it's also good for beginners because they have such a wonderful, website where they have very, very clearly explained tutorials. And I really love it. And I think it should be the go-to resource for everyone. Like, I wish I had started with Giotto, really. But the thing about Giotto is that not only do they have the basic steps, they also have a lot of practical tools like things like binning size. So for Visium HD, many of you would probably have that question in mind, 2 micron or 8 micron or 16 micron, right? So I think Giotto actually tackles all these questions that has to do with spatial, which I don't find in Surat or in the SCVerse websites. So that would be a really good suite for everyone to take note of.
Has like a cell segmentation with like the H&E images, or Is
I
it just
cannot-
like
Giotto,
play
right?
with, yeah, play with the bin size?
You play with the bin size. Yeah, that's what I remember. Yeah, unless I miss something. Yeah, but that was such a wonderful thing to just, yeah. And you can define your bin size. Yeah, but they do it in hexagons. Yeah, yeah. They have these hexagons, and then uh, but basically your parameter will be how many hexagons do you want? Yeah. So I thought that was very cool, actually. Yeah. Okay. Yeah. Any questions? Okay. And also, these are not limited to only Visium HD, right? So if you're coming here, we are teaching you Visium HD, but think broader than that, that Visium HD is a spot-based platform, and all the principles and everything that you learn is going to be applicable to any kind of spot-based, right? It's just that the variations between the platforms will kind of like matter when you get down to the granularity of the analysis tools that you use. Okay, so there are many methods that are under development, and there is a lot out there. Every day there's something coming out. I'm not able to keep up. So if I miss out anything, I apologize. But at the end of the day, I think going back to the basics, I think it's all about understanding your platform, your question, and what kind of resolution you want. So everyone talks about subcellular resolution. So the Visium HD is 2 microns, Studiosig is about 250, slide tag is about 10 nanometer. And then there is sensitivity. The more you aggregate your bins, the better signal you're going to get. But then you are also going to be losing, all right, some aspect of your cell-cell communication, your so-called single cell, right? But the question is, do you really need it, right? And then there is also algorithmic decoding. So when you look at imaging-based, each of these imaging-based single molecular platforms, they have their own decoding, encoding, and decoding. And we might not appreciate it much because many of it is not open access, is that these algorithmic decoding matters because the probability of what a spot is and whether that spot is a gene matters, especially in the context of how dense your tissue is and the type of tissue. And then the definition of what the simplest unit. So spot versus single cell, the type of QC, the extent of pre-processing. And one thing that I think we will broach this during the workshop is the concept of counts. The concept of counts in a spatial setting is very different. So you're talking about Poisson distribution of your transcripts. And what you define as minimum number of genes and minimum number of counts, all right, is going to be very different. You're going to lose, it's very easy to lose cells in spatial analysis as compared to actual single cell. But that's because we're also amplifying more in many of these single cell platforms. We have not hit that sort of strength in many of these spatial platforms. So we need to be aware of all these. So yeah, so at the end of the day, all of these factors is going to affect how you're going to answer. And then segmentation, yeah, I know you always, so I like segmentation, but that's because I'm working on the Xenia, right? So I do segmentation and I do a lot of images. But whether it is spot-based or whether it is single molecule or whether you're not going to be doing transcriptomics, the problem is the same, right? Segmentation, you're drawing boundaries. And it is true, we don't have that depth. 5 micron, there are people who will come here and they will fight you, all right, that 5 micron is not single cell. That 5 micron is just going to give you a snapshot, right? So it's not a droplet based. You're not there actually merits to dissociating your tissue, right? You're actually dissociating them into single cells, encapsulating them into drops, right? And each cell is basically a cell. But in this case, it's not. It's just a snapshot of a cell. It's already been sectioned off. And you have cells of different sizes. So just think about it. When you normalize, you are making the assumption that each cell, you have a scale factor, but your cells are all chopped off, which means that you're not even getting a true representative of your transcript counts or your gene numbers. And all this adds on, and what adds on further is that we now know, as we learn more, is that genes are not even evenly distributed. Sometimes they are localized to a certain aspect or to a certain geometry or point, right? So think about all of this. So what I'm saying is that we have to borrow the ideas and the methods that were developed for single cell, and we need to apply it. And that is logical to do. But just don't fall into that trap that it is a direct, implementation, and everything is just a linear mapping from single cell to spatial. Okay, so wet lab and engineering. So yeah, so a tremendous amount of time, effort, and creativity and intellect goes into all these development, and we don't talk about it enough. All right. So before we even have this workshop, before even we have a Visium HD or a StereoSig, you have the bench work, you have the engineering work. All right. And the value of what we analyze is as good as the input that we provide. And after our computational analysis, there is a whole few months of wet lab developments and projects that are born out of this. All right. So it is a two-way process. It's not that one is more important or less important. I think it is something that we have to understand. It's a learning, it's a two-way thing. And that is why it's important I think it's important for those of us who are doing wet lab. I think it's important to understand the computational process too. We need to appreciate it. I think it also goes the other way around, that we need to appreciate the bench work that goes into it as well. So I highly recommend reading these papers, materials, and methods. So this is a 2013 paper. And I believe this group formed Katana, which is a Swedish company, which actually set the whole in situ sequencing technology that was later acquired by 10x Genomics that laid the foundations for the Xenium. If you look at this first paper, and don't read the introduction and the results and the conclusion, please go and read the materials and methods, all right? And these are the materials and methods. These are the headers: cell culture and sample prep, tissue section, in situ reverse transcription, gap fill padlock probing, barcode padlock probing. Then there's RCA and RCA products detect, sequencing by ligation, image acquisition and analysis, all right? So a lot of thought that has gone into coming up with all those nicely packed mixtures that you use for your spatial transcriptomics experiments, right? And if all of those enzymes and buffers were out of order, we would not have the signal that we are getting right now. And the thing is, it is important, I guess, to understand this, because I think the computational analysis is going to now drive forward further development, and then it's going to go back into a cycle. So take your garbage in, garbage out seriously, all right? Because you need to ask back and forth questions. And it's also something that I'm learning to do as well. And that is how we advance forward. So what this workshop will teach you and your approach to this course. So yes, this workshop is computationally heavy. We are going to be doing more hands-on on your laptop. It's only module 1 that is wet lab. There are things that we cannot control. So things like getting everyone into a wet lab is administratively, it's not feasible. So we've tried to give you the best, and you have really really, really wonderful instructors, Amina and Melanie. All right, they know their stuff, so engage with them. Now, modules 2 and 6 will be computational. Now, the computational practical is foundational. I know that all of you have different, have different levels of experience, right? And we don't expect everyone to know everything because everyone has got their own niche. And we are also, again, limited by computational bandwidth. These data sets are not easy. All right, so we've tried our best to balance it, given the restrictions, given the limitations, and also to cater to the mixture of participant cohort. But we are all here. So if you have any questions, all right, or you have any doubts, or you need anything that is more personalized, just come and find us. All right. And then, yes, we are focused on the Visium PhD. It is spot-based and it's from 10x Genomics. But like I said, if you have any questions of other spot-based platforms, please come and ask us. And many of these concepts are broadly applicable, except for some finer details, all right, that will be different. So we don't want this to be just you coming and listening and clicking on the what do you call it, the go or the signs on your RStudio. Think critically at every step, right? At every code, right? Think about your project. Think about how it's applicable. And if it's not applicable, then ask yourself why. And you can come and ask us, oh, this is not applicable. Like, what can I do? I don't think this is helpful. I know all this is important because sometimes knowing what doesn't work for you is just as important as knowing what works for you. And even though the analysis might not be challenging at all times, but there'll be discussion points. So participate and brainstorm, and no question is like too small. So in the end, so I guess because in the end, it is not the brand, it is the idea, skills on how you approach spatial problems that will be the take-home points from this workshop. So really, really, the world is your oyster. There's so many out there. And there are now active research in plant and insect, analysis, medical applications, and of course, there's virtual and your augmented reality platforms that are coming up, Apple Vision. So yeah, well, I mean, it's too expensive, but it's very cool, right? So yeah. So with this, I conclude and I want to welcome everyone. And I really want to thank my co-organizers from Bioinformatics.ca, Nia Hughes, and from BSCI, Urja. Urja is here? No? Oh, she just stepped out because she knew that I was going to mention. And then my instructors, our instructors and TAs, all right, who really make this whole thing happen. Savannah Kilpatrick, Alyona Ivanova, Amina Abu, Melanie Peralta, Joanne Kan, Yen Wu, Javaria Atif, and Dina Karambuk. Am I pronouncing it correct? Okay, all right. And that's it. Oh, and then, yes, about panoramics. So we are a pan-Canadian working cluster for spatial and single-cell omics. We are going to have upcoming single-cell and spatial programs. We'll be updating it in coming weeks and months. We just had a single-cell controversies and challenges in dimensional reduction in single-cell technologies. So we're going to have more spatial stuff. We just need to get timings right, because everyone is too busy. And all our events fall under the Inspire, Enlighten, Challenge Me chapters. All right. It is what they say. Inspire is really to inspire you to think out of your box. Enlighten Me events are all workshop settings, which we do mostly in collaboration with Bioinformatics.ca and similar organizations. And Challenge Me is really to challenge you to think beyond what is traditional. So yeah, so if you want to know more about panoramics, this is the QR code. If you want to ask me any questions, you can also scan this QR code here, or you can email me.